So, when do we use PCR? Well, I'm going to go through three major applications. The first one is going to be screening PCR. So this is to check whether your expected sequence is present. This can be used for a number of purposes. In your synthetic biology projects in iGEM, what you'll probably be doing is checking to see if your insert is present in your colonies. So that would be screening for colony PCR. But it's also used in the real world quite a lot. So, for example, forensics screens for certain nuclear elements that are repeated. I forget the name, but they use PCR to search for small elements. And PCR is used because it only requires tiny, tiny amounts of DNA. Additionally, there's companies that use screening PCR on samples of your gut microbiota, and they try to see which bacteria are present. And depending on what bacteria they see, they can tell you, oh yes, your gut has this profile. So it can be quite useful. But let's discuss it in terms of iGEM. So you're going to be making plasmids. Well, you're going to be taking plasmids and you're going to be assembling inserts into them. So our inserts in red. But how do you know that the correctly transformed bacteria actually have the insert in? How do you know it's not an assembled plasmid? Well, the first answer is do it with Gibson or Golden Gate because they have very high yields. But that's not a particularly robust answer, and that's not an answer that the scientific community will accept. So we use screening PCR as our first line of checking. The gold standard is sequencing, but sequencing costs more. So there are multiple ways that you can do screening. You can pick primers that go around your insert. And that's quite useful because that will amplify the entire sequence. And if you do that with a high fidelity polymerase, you can actually send just the insert off for sequencing, which could be cheaper if you're not interested in sequencing the whole plasmid. An alternative option is you can put primers just inside the sequence. In fact, yes, so both, in this case, both of them would be just inside the sequence. This is the wrong color pen. And in this case, you would then run the result on a DNA gel to check for length. And if the length is what you expect, then you can say, okay, my insert is in there. And otherwise, you wouldn't see any amplification. And then there's a third way. And this way is particularly important with restriction, digest, and ligation, because that can produce wrong orientations, which is where you place one primer outside of the insert, that way, and one inside. And this should only amplify if the insert is in the correct way, because if the insert is in the wrong way, then the insert specific primer ends up ligating annealing there, and that's not amplifiable. There's no amplification that will occur. So you can run it on a gel, and if you see nothing, then you know either there's nothing in there or its orientation is wrong. And if you combine that with another screening set of primers, then you can say, oh, okay, either the insert's not there or the insert's in the wrong orientation or the insert's in the right orientation, which would be good. I sincerely hope that happens for you. So screening PCR normally takes Taq polymerase, which is low fidelity. And the reason we do that is because you don't need the expensive stuff. All you're looking for is length. If you're sending it off for sequencing, you would need a higher fidelity polymerase. So that brings me on to my second use of PCR. This one is amplification. The reason I say that brings me onto this is because amplification is where you have a genome or some sort of template DNA, usually from an organism, and you have template DNA. And you want to get the template DNA amplified, just that amplicon, so that you can use it for cloning. And the reason I said this twice now, the reason that I said it leads me on is because amplification takes a different polymerase. It takes a higher fidelity polymerase, usually Q5, which has about 1,000,000-fold higher fidelity than TAP, which means it makes 1,000,000-fold less errors, which is pretty good, especially when you want your sequence to be perfect. So amplification is quite simple in theory. Your primers just sit on either side of the sequence you want. They amplify it up. Bob's your uncle, you've got it. However, since amplification is usually for cloning, we have to analyze what the primer sequences actually look like. And it tends to be that you need to insert on either side of the plasmid extra bits that help with cloning. So if we take this primer here and expand it, what we actually want to see is the annealing region as normal. That's just a normal primer for amplification through PCR. And then we would include a site. So if this was for a Golden Gate purpose, what I would expect to see is my 4 base pairs that would be complementary in black, and then my BSA1 or whatever restriction enzyme I'm using, I'd see the type 2S restriction enzyme recognition site. And I would then be able to assemble that into an appropriate vector. Alternatively, I might look at Gibson. And if I was looking at Gibson, then I would expect to see a 25 base pair homologous region to my plasmid that I'm going to assemble it into, not to the genomic DNA. So what this actually looks like on the genomic DNA is the rest of it just sits up there. And when you run the PCR, as it's running, this bit then starts to get amplification onto it as well. So all your amplified bits have that extra bit. And the reason you keep that tail and not the tail of the genomic DNA specifically is just because your primers are really high concentration and all the new DNA is made from the primer. And next to no DNA is made from the template strand. So the DNA that's made from the primer has to include all the primer bits. When I say made from the primer, I mean extended from the primer. So that should hopefully make sense.